Okay, so this week we're going over photosynthesis. We're going to talk a little bit about the organisms that undergo photosynthesis and why they are so important. And we're going to talk about the process of photosynthesis itself and the organelle that is actually doing the photosynthesizing. Now, as we go through this, I would like you to think about respiration, what we covered last week in the last module, and kind of compare and contrast respiration with photosynthesis. In many ways, respiration is going to be the reverse process of photosynthesis. With photosynthesis, we are creating glucose, while in respiration, we are breaking down glucose to release energy. Okay, so life on Earth can really be broken down into two categories, either heterotrophs or autotrophs. And when we're talking about organisms that can undergo photosynthesis, they fall under the autotroph category, where autotrophs are organisms that make their own food. So specifically, what's being made in photosynthesis is glucose. So that is the food source where energy is going to come from. And these organisms are going to make that glucose, then store it, and then use it later to create energy to do some work. So specifically throughout this PowerPoint, we're going to focus on plants, but it's important to understand that not all photosynthetic organisms are plants. There are plenty of organisms that aren't plants that can undergo photosynthesis. And now compare that to heterotrophs. So heterotrophs can't make their own food. We as humans fall under this category. So we need to eat our macromolecules and to gain energy from our food. Okay, so looking at autotrophs just a little further, and if you break down the word autotroph, it tells you everything you need to know about these organisms. Auto meaning self, troph meaning nourishment. So these organisms are making their own food. And they're doing this from inorganic substances. So, with photosynthesis, what we are doing is taking carbon dioxide or CO2 and water and using energy from light to combine the CO2 and H2O to form glucose. Now, you might be wondering, how is CO2 not an inorganic substance? Well, that's because by definition, an organic substance is a carbon-based molecule that has a hydrogen attached to the carbon, or at least one hydrogen. So with CO2, there's no hydrogen. And that is where water comes in. So water is where the hydrogens are going to come from that will later attach to the carbons to create these glucose molecules. Now, there are a lot of different types of autotrophs out there, but most of them are photoautotrophs, meaning they're going to use sunlight to create glucose. While there are some species of organisms that can undergo autotrophy using chemical means, but these are pretty rare. Most autotrophic organisms are going to use sunlight. So again, with photosynthesis, what we're doing is taking carbon dioxide and water, H2O, and combining those two things to form glucose molecules. And there's really two products to photosynthesis. Yes, we end up with our glucose, our organic molecule at the end, but we also have oxygen as a byproduct. So O2 is going to be released from this chemical reaction. And that is where a lot of the O2 in our atmosphere comes from. So without these photoautotrophs, we would not have the atmosphere that we have today. And many of the organisms can't survive without this oxygen in the atmosphere. Okay, so next let's compare respiration and photosynthesis and just take a look at their chemical reactions. So in this blue box right here, we have respiration. And if you notice, look at our reactants, our starting reactants. We have one glucose molecule and 6 oxygens. Remember, it takes oxygen to undergo respiration. And through this chemical reaction, we're going to store energy in ATP. Remember, ATP is how cells store energy, and they'll use this ATP to do some sort of job or function. And then the products of this reaction are 6 CO2 molecules and 6 water molecules. And whenever we breathe out CO2 because our bodies have undergone respiration and we need to get rid of that CO2 somehow. So when we exhale, we are releasing that CO2 into the atmosphere. Now compare that to the photosynthesis chemical reaction. So our starting reactants are 6 carbon dioxides and 6 water, and then we use light energy to convert this CO2 and H2O into a glucose molecule and 6 oxygen molecules. And if you look at these two compounds, they are both balanced in the same way, but they're really just the reverse of each other. With respiration, you start with a glucose and 6 oxygen, while in photosynthesis, you start with 6 CO2 and 6 water molecules. With the respiration, we end up with 6 CO2, 6 water molecules, while in photosynthesis, we end up with 1 glucose and 6 oxygen molecules. Okay, so where is all of this taking place? Well, we're going to focus on plants in this PowerPoint. And remember, plants are eukaryotes, meaning they have membrane-bound organelles. And if you remember last week when we were talking about respiration, the membrane-bound organelle that was used for respiration was mitochondria. Well, this week with photosynthesis, we have a different organelle that is going to undergo photosynthesis, and that is called a chloroplast. So a chloroplast is an organelle that is found inside plant cells that allows photosynthesis to occur. And looking at this picture down here, you see a plant cell. So I'm outlining the plant cell here, and you could see all these little organelles floating around it. These are your chloroplasts. Now, these plant cells have other organelles than just chloroplasts. However, you can't see them. We only see the chloroplasts here. And that's because organelles are usually transparent. But chloroplasts aren't because they're full of photopigments. They are pigmented. So they have this green pigment that is there that allows us to see these organelles under a microscope. Now, there's many different types of photopigments, but the most abundant one is chlorophyll A, and chlorophyll A actually reflects green-blue light. So let's take this chloroplast right here as an example. So this is a chloroplast. This is one organelle. This is found inside the plant cell. Here in the yellow is the membranes of the chloroplast. So you have two membranes for the chloroplast, just like we saw in the mitochondria. And then inside the chloroplast, we have a third membrane that creates stacks or discs. We refer to these as thylakoids. All right, so these thylakoids come in stacks, and this is where photosynthesis is going to occur. And this is where you're going to find those photopigments. Now, when light enters this chloroplast and hits this thylakoid, the photopigments are going to absorb many of the wavelengths of light and use it to create energy to form glucose. However, it doesn't do this for all the wavelengths. Chlorophyll A reflects green-blue. So those wavelengths, the greenish blue color, is going to be reflected or bounced off these thylakoids, and this is what hits your eye. This is why plants look green, because the green wavelength is the one that's making it to your eye, while the rest of the wavelengths are being absorbed by the thylakoids. Okay, so we have more than just chlorophyll A in plants, though. So with photopigments, we can classify them as two major groups. We can either classify them as primary pigment, which is your chlorophyll A, or we can classify them as accessory pigments, also known as secondary pigments. So for all photosynthetic organisms, chlorophyll A is going to be your primary pigment. It is going to do the work of photosynthesis. And just like we talked about in the last slide, chlorophyll A is more of a blue-green color. So it's going to absorb all the wavelengths of light except for blue-green. It's going to reflect that color. However, there's still energy in these wavelengths. It's just chlorophyll A can't harness them. So to help chlorophyll A, we'll have all these secondary pigments. So here's just a couple of them that plants can have. Many plants have chlorophyll B, which reflects a yellowish green color. So it's not harnessing these wavelengths, but it is harnessing the rest of the wavelengths. Specifically, it's pretty good at harnessing blue light, which chlorophyll A could not pick up. Then we could also have xanthophylls, which are more of a yellow color, again, reflecting yellow, absorbing the rest of the wavelengths, again, picking up the slack of chlorophyll A and chlorophyll B. And then we could have carotenes, which are more of a yellow, orangish, and red color. So they're reflecting these colors, but absorbing the rest. So with these extra accessory pigments, we can absorb the blue-green wavelength. So that's how these are helping chlorophyll A. However, chlorophyll A is the center of the photosynthesis. So whenever these other photopigments pick up light energy, it's going to pass that energy off to chlorophyll A, where work is going to be done to create glucose. Now, if you've ever been up north during the fall, you might have seen the leaves change. And what is happening here is as we approach winter, the sun gets lower in the sky. And as the sun gets lower in the sky, chlorophyll A becomes less efficient at absorbing wavelengths of light, and the other photopigments become more efficient. So during summer and spring, when the trees are green, the reason they're green, even though they have these other photopigments, is because they have much more chlorophyll A. It is a much more efficient photopigment, so you want a lot of it. So they look green even though they have these other photopigments. They're just masked by how much chlorophyll A is present in the leaf. However, as winter approaches and the sun gets lower, chlorophyll A becomes less efficient. So it doesn't make sense for the plant to keep making chlorophyll A. It'll actually break it down and rely on the other secondary pigments for photosynthesis. So as we lose chlorophyll A, we'll start to see more of the chlorophyll B and the xanthophylls and the carotenes. And so the leaves start turning more of a yellowish, orange, or red color. So that is what's happening during fall. We are losing the efficiency of chlorophyll A, so we start removing chlorophyll A, losing that green color, and the other photopigments start to appear. Okay, just to reiterate this further, we have all these accessory pigments because they pick up on wavelengths of light chlorophyll A cannot. So this is typically what a photosystem is going to look like. You're going to have all these secondary pigments surrounding the primary pigment, which is chlorophyll A. So we have all these secondary pigments that are picking up different wavelengths of light, generating energy from it, and then passing that energy off to chlorophyll A, where photosynthesis is going to occur. So we can actually see this when we look at this graph. So we can measure how much absorbance is happening with these different photopigments. So with chlorophyll A, you'll see the absorbance of different wavelengths in this gray line here. All right, and this is the different wavelengths of light. So where the highest absorbance is occurring is where most of the energy is being generated. So you see that we have a high peak right here, and we have a high peak right here, which is more in the purplish color and the reddish color, reddish orange, that is generating a lot of the energy for the plant. And notice where the least amount of absorbance is occurring. You might have been able to guess what color this is going to be. So this is going to be green. And the reason it's so low in absorbance is because it's not being absorbed by the chlorophyll. It's actually being reflected by the chlorophyll. And that's why the chlorophyll appears green. Okay, so we have this wavelength here that chlorophyll is not doing a very good job of absorbing. That's why we have these secondary pigments, because they could kind of pick up the slack here and generate some energy from this spectrum right here. Okay, so going into this even a little further, you might remember from other science classes that white light, the light that comes off the sun, is made up of all the color wavelengths. So we have all the wavelengths of light inside white light. And if you pass white light through a prism, you'll be able to separate out each one of these wavelengths and see the different colors that are there. So there are three things that happen when light hits an object. All right, so when light strikes an object, it can either be absorbed, which if it's the chlorophyll, this is what's going to be used to create energy, the light that's absorbed. It can be transmitted, which means it passes through the object. So some light can squeeze through or pass by this chlorophyll, or it could be reflected. And with chlorophyll A, it's more the green blue that's reflected, which is why plants look like a greenish color. So just to see this even further, let's look at these three boxes and see what's going on with the light here. So whenever you look at a black object, like this black box right here, what is happening with the light? Why does this appear black? Well, that's because all the wavelengths of light are being absorbed by this black box. So there's nothing reflecting off of it and hitting your eye, which is why it doesn't look like a color. Compare that to this white box right here. So whenever we see a white object, it's because all the wavelengths of the light are reflecting off this object and hitting your eye. So you're not seeing individual wavelengths, you're still just seeing the white light that's bouncing off this object. So nothing is being absorbed, everything's being reflected, versus the black box where everything's being absorbed, nothing's being reflected. And then compare these two to this green box. So what's going on here? Well, the reason this box looks green is because this box is reflecting green light. Green light is bouncing off this box and making it to your eyes, and that's why it appears green. However, it's absorbing the rest of the wavelengths of light. So it's taking in the rest of the wavelengths. Okay, so how could we actually see these different photopigments in plants? Well, one common way to do that is this scientific method called paper chromatography. And what you're doing with paper chromatography is really separating out the different molecules based on their characteristics. So how this works is that you would use a polar stationary paper. So this paper right here is polar. And what that means is it has a slight charge. It has a polar charge to it. And you would stick this polar paper in a nonpolar solvent or nonpolar liquid. So we'd put this paper in a liquid that's nonpolar, usually an ether-based chemical compound. So the liquid has no polarity while the paper has polarity. And what we do is add a little bit of ground up leaf from the plant at the base of this paper, stick it in this liquid, and let the liquid creep up the paper over time. Okay, so what is going on as this liquid is creeping up this paper? Well, as the water or the liquid rises up the paper, it's going to take the photopigments with it. And any of the photopigments that have a lot of polarity are going to stick to the polar paper. Now, remember in chemistry, like likes like. So these polar compounds are going to like the polar paper and stick to it more. So they're not going to travel as far. Versus the nonpolar compounds. The nonpolar compounds are going to stick with the nonpolar solution or solvent. So this solvent is creeping up this far, so all the nonpolar compounds are going to be with the solvent line. So as far as the solvent goes, that's how far the nonpolar compounds go. So what we're effectively doing here is separating out these different pigments based on their polarity. All right, so in chemistry, there's a general rule to follow. The more OC bonds that you have, the more polar a molecule is going to be. All right, so as we looked at this paper chromatography, we had polar paper, nonpolar liquid that is going up the paper. If things stick to the paper more, we consider those to be in a stationary phase, while if they moved with the liquid more, we call them to be, or say to be more in a mobile phase. All right, and whether a molecule is in a stationary phase or a mobile phase depends on how many OC bonds it has. So look at these photopigments right here after paper chromatography was conducted. Can you tell which one's going to have more OC bonds, going to be more polar? Well, you might have been able to guess it that chlorophyll B is going to have more polar bonds. It's going to have more OC bonds. And that's because it went up the paper the least. It's sticking to the paper more. Remember, the paper is polar, and so this pigment is polar and doesn't travel as far. Compare that to carotene, which is more nonpolar. It has less OC bonds. It traveled the furthest. So carotene's more in the mobile phase because it did not stick to the paper because it's not polar. It's more nonpolar. So it's going to travel with the nonpolar liquid that is traveling up this paper as well. Okay, so with paper chromatography is just one way we could separate out these compounds and see each of the photopigments individually. All right, so what do we do with this information? Well, we can compare photopigments by calculating something called a retention factor. And retention factors are really easy to calculate. What you would do to calculate a photopigment's retention factor is first measure how far the solvent went up the paper. So if this was the starting point right here, and the liquid traveled this far, then the solvent traveled exactly 10 centimeters. And then you would measure how far each of the photopigments traveled. So again, we'd measure from the starting point to how far each of the photopigments traveled. Just like here with the yellow, it traveled about 2 centimeters. So for the retention factor, all you would need to do is divide the distance the pigment traveled by the distance the solvent traveled. So for the yellow pigment, its retention factor is going to be 2/10. Now compare that to the blue pigment, whose retention factor is going to be 8/10. Okay, so just looking at retention factors, can you see which one's more in a stationary phase versus a mobile phase? Well, if it's a smaller number, just like the yellow is, it's going to be more in the stationary phase. While bigger numbers or numbers closer to 1 are going to be more in the mobile phase. And now remember, the paper itself is polar, while the solvent is nonpolar. So if it is closer to 1, just like the blue pigment is, it's going to be more nonpolar molecule versus the ones that are in the stationary phase or didn't move as far, they're going to be more of a polar molecule. So just looking at retention factor numbers, you can tell if a molecule is more polar or nonpolar. Okay, so we've talked about photosynthesis, we talked about the products it makes, we talked about the reactants it takes to undergo photosynthesis. So we know plants undergo photosynthesis, but do plants also respire? And the answer is yes, they do. So plants also have mitochondria. They have chloroplasts that undergo photosynthesis, but they also have mitochondria that undergoes respiration. So the plants are using everything made in photosynthesis for respiration, which you can see in this cycle right here. So these aren't different cells, these are different organelles. So both these organelles would be in the same cell right here. Here, the chloroplast is undergoing photosynthesis, where you take 6 CO2, 6 water molecules, and you create glucose and oxygen. And then that product is passed down to the mitochondria, where it's going to use that glucose and that oxygen and create CO2 and water and store energy as ATP. So plants, just like any other organism, have mitochondria that is going to store energy in the form of ATP from glucose molecules. So this CO2 and H2O can then be passed back to chloroplasts where photosynthesis can happen again. Now you might be wondering if chloroplasts and mitochondria are really the reverse of each other. And everything that's made in a chloroplast is being used by the mitochondria, and everything made in the mitochondria is being used by the chloroplasts. How is there enough oxygen in the environment for the rest of us if plants are doing both these things already? Shouldn't they cancel each other out? Well, the answer to that has to do with how quickly each one of these processes occur. Photosynthesis is a much more efficient reaction than respiration is. So photosynthesis happens much quicker. So you get this surplus of sugar and O2 inside plants. They don't use nearly as much as they produce when they undergo respiration. So you have this surplus of these products and you make them faster than you use them in respiration within plants. And they make so much of it that they have enough left over for the rest of us to survive as well. And it's really only at night that you start to see the difference. In different environments, or even if you did this as an experiment in a lab, you'd see a drop in oxygen at nighttime. And that's because plants are still undergoing respiration at night, but they stopped undergoing photosynthesis since there's no sunlight. So if there's no O2 being formed, but respiration is occurring in plants, you're going to see that drop in O2. Okay, so that is everything we wanted to talk about in this PowerPoint. Hopefully this gives you a good understanding of photosynthesis and it will help you with the rest of the module. So again, we just really hit the major topics in this PowerPoint, and it was more of a guideline just to help you understand more of the nuances in the module itself.